Hi, everyone. My project this summer has been on investigating strain colonization in the human gut microbiome using in vitro communities, and I've been working on this project with Casey Huang's lab and David Relman's lab. So the human gut microbiome is an extremely diverse, highly individualized, and generally very stable community of microbes that's present in each person's gut. And there's a lot of evidence that the diversity of the gut microbiome is an extremely important part of human health. This diversity can be impacted by a lot of different perturbations. For instance, it can be significantly reduced by antibiotics, but there's also a lot of evidence that it can be maintained or even increased by colonization by new microbes, which can happen either naturally from the contact with other people or with the environment, or can happen by more targeted interventions like probiotics. But right now, it's really hard to predict the success or the specific effects of colonization in the gut. There are a lot of unanswered questions like how well will an introduced microbe actually be able to colonize? If it is able to colonize, how long is it going to stick around for? And how will this affect the rest of the species in the microbiome? And so this summer, I've been working on trying to answer some of these questions using in vitro communities. So the main advantage of in vitro microbial communities is that they're reproducible and manipulable systems in which we can perform various experiments involving mixing communities and adding specific strains to communities in order to study colonization in a much more controlled laboratory context compared to the more complex environment of the gut microbiome. So at the beginning of this summer, we generated 192 communities from human stool samples. And so in order to do this, we basically just inoculated the small fragment of stool into liquid media and let the bacteria from the stool, which are from the gut microbiome, grow in this media. So in order to use these communities for future experiments, we needed to make sure that their species level composition was stable and not changing significantly over time. And so what we did was we passaged these communities several times. And so what passaging is basically after the bacteria from the stool, after the bacteria in the communities grows up for 48 hours, we dilute the saturated cultures at a 1 to 200 ratio into new media in order to let the bacteria continue growing. So we hypothesized that there would probably be a lot of shifting in the community abundances right after the communities were inoculated into media from the stool samples, because it's just a very different environment. And so we passaged these communities 7 times. We estimated that, that would probably be enough time for the communities to reach stability. But this is a question that we want to answer because we're not entirely sure. And so we performed 16S rRNA sequencing in all of our communities, and we're currently in the process of analyzing the data that we've gotten in order to answer this question and other questions. One of the other questions we also want to answer, for instance, is how similar are the communities at the end of the 7th passage to the starting stool samples and to the first few passages? How much change has there actually been over time over the course of these 7 passages? We want the communities to be as similar as possible to the stool samples in order for them to be the best system for studying colonization in the gut microbiome. And so we also decided to set up a second set of the same communities and passage them a few times at a 1 to 6 ratio. And so we hypothesized that doing this lower dilution might actually help to preserve slower growing species and make the final communities look a bit more like the initial stool samples. So hopefully from these 2 sets of communities, we'll be able to learn a lot about the stability of in vitro communities and how suitable they are as a system for studying the gut microbiome in general. So now I want to talk about one of the major experiments that we did this summer, which was to mix communities at different ratios. And so one major question we had about colonization was how many microbes would actually need to be introduced to a community in order for colonization to occur. So we set up an experiment to test the effects of mixing microbial communities at different ratios. We took 8 different communities and we mixed them at 7 different ratios, from like 1 to 1,000 to 1 to 100 to 1 to 1, and the other direction 1,000 to 1. And then we passaged each of these mixtures 5 times. And so the first question we asked was basically, how does the ratio at which communities are combined affect the final community state? Does the final mixed community look more like the introduced community if you give the initial community a larger inoculation dose of the introduced community? And then another question we asked was, what makes certain taxa more successful colonizers than others? Are there certain taxa that are able to colonize just as well if you give only a small inoculation dose compared to a large inoculation dose? And what makes these taxa better colonizers? So hopefully this experiment can help to answer those 2 questions and clarify the effect of inoculation dose on colonization success more generally. Another thing I wanted to talk about was our strain isolation. So some of our future experiments will involve adding single strain isolates to communities in order to investigate the dynamics of colonization. And so this summer we worked on isolating strains from our communities for use in those future experiments. So far we've isolated on 5 different types of media, both very general media that contain a lot of different nutrients and support the growth of various taxa, and much more selective media that have fewer nutrients and even in some cases contain antibiotics and are only capable of supporting certain taxa of microbes. So far we've isolated from 3 of our communities and we've cultivated a total of 17 unique species from 8 families. And we're hoping to continue isolating from more communities and perhaps more media types to build a much larger library of strains, which we can then use in our future experiments. So overall this summer, we generated a large set of communities from stool samples, and we began to investigate colonization by performing community mixing experiments. And so our next steps are going to be to analyze the data that we have from these communities and experiments, and to perform more experiment mixing communities and adding strains to communities to continue to answer questions about colonization. Thank you.